Hello. Hi there. I wanted to go through a few questions that came up this week and then go through the Unit 5 content area. I've had a few more questions on grades. So I wanted to point out there on your grades that you have two bars you can look at. One is class progress. That is not necessarily your grade. So this just tells you how much you've done in a particular area. So like this is a sample student. So this student is way behind on probably viewing the content files and any video. These aren't actually any assignments, but just views that are in there. And so you'll see as you go further along, this isn't necessarily my grade, it's just how many items that I've worked on there. So it gives you, 100% of the quizzes have been done. This is a practice student, so all the grades have been entered. And a lot of this you can just ignore. There may be, I don't have it in my view here, I do as an instructor, but I'm actually able to customize. I have a little settings icon and I can actually take some of these items out. But just know that, there probably are a few things down in here, like login history is probably still good, but like surveys, checklists, that you can just ignore. So if you don't see anything happening in those bars or even see a bar there, that means that there aren't any items even to complete. Now, when you look at the grades, I'm still getting some questions on dropped grades. I know that's alarming to see that kind of red dropped. Remember that it is dynamic. So some areas there are no drops available, like the week one. Those were those first three assignments you had to complete. But like practice quizzes, final quizzes, and your study guides, you get 2 items dropped. And so this is a sample student. I've given them full points on every single item. But let's say, week 8 or unit 8, you forget to turn it in and you get a 0 here. What is really nice is that this dropped will now move down to that 0 after that 0 grade is assigned. So you can see right away, oh, did that impact my grade? Did that not impact my grade? If you're within your 2 drops, you'll be okay. Or maybe you just scored really poorly on a final quiz. You'll be able to see right away that it's dropped. So it's just more transparent. I realize it's a little different. Blackboard was doing the same things, but they just didn't mark it to the student as dropped. It was reflected in the total, but maybe wasn't so obvious. So same thing with the study guide. So just don't be alarmed by those. And this will move and change. So, once the grade's been assigned, obviously if you've missed the assignment, I need to go in and assign the 0 first, but you'll see that drop to move. Okay. Another interesting thing that came up this week when meeting with a student is I know a lot of you are really carefully watching to make sure you get check marks over here. And you can see I'm not a very good demo student because I haven't taken all the quizzes, so I don't have any checks. But if you are going through and you're not getting that, make sure, like here, I don't have it because I haven't turned anything in. But let's say you turn it in and you should see a check mark here for some reason and you don't. A couple things to check. There are a few areas where you can actually check it yourself when you're in there, and you can tell that by, before I launch that, hopefully I'll get a screen there. Um, but there'll be a little box underneath to check it, or I can just check it out there on the main page. Yeah, this one I would have to check out on the main page. There we go. So I could check that it was completed. Another important thing, if you're not getting checks, instead of using these breadcrumbs or going to the back arrow on your browser, which is usually the way I navigate things, make sure you're clicking these arrows to go through the material. So you're turning it in and this is just tasked as view the topic, you're turning it in and you're going forward through these arrows, and then that should flag it. So if you're submitting especially like these virtual labs and you're not seeing that getting checked off, you may just actually need to open this page, not necessarily redo the lab again because you've already done that, but just scroll through this to mark that you have viewed those. So that's probably what it is. So I went through that with a student this week, and we both discovered that that seemed to work. And I think that's about it there. So I'm going to go ahead and move on to Unit 5. I do have a lot of students that are working ahead, which is fine. So keep in mind the midterm is only going to be open specific days. So if you, finish early, you may just end up with more study time to complete there. All right, so let's go. There's only one content file for Unit 5, and there are no virtual labs. So we're a little bit kind of not going to be doing the virtual lab, so taking a break from the virtual labs. There's a few other simulations that we might do out on the web, but in the McGraw-Hill material itself, It's going to be a few weeks before we do those again. So don't be alarmed, those aren't there. It's just because the virtual labs don't match the learning objectives. We have very specific objectives set by the state and for transferability and to work into all the clinical programs. So if there isn't a virtual lab for it, we have to come up with something else. So if you're not seeing that, that just means there wasn't a good example for that for us to use. Okay, so this one just has the content file and just one. So there's a little bit shorter this week, so it's a good week for you to maybe work ahead a little bit if you've got some busy weeks coming up. The big piece of this unit, even though I know students spend a lot of time, trying to identify cells and looking at all the differences, but the big piece is really the dichotomous key. So a dichotomous key is just simply a way to break things into components. So, um, this system's used all the time. So it can be used for, more formal scientific things like identifying rocks or birds or different plants or things like that. So it's kind of like 20 questions if you've played that, is it a plant or is it an animal or is it alive, living or non-living? So it's a way to sort of, eliminate a whole group um, by a set of questions and looking at features, and then get that group that you do have and narrow it down as you go. But this is used, I mean, you think about it like, really to apply to even set up like the layout of a grocery store. Like, okay, is it a canned good? Is it not a canned good? You kind of go through those questions, just stocking things and thinking about where things go all the time. So we are going to be using these quite a bit in the lab. So this lab is really about starting to practice those. So the beginning portions of the lab are just kind of going through the samples. So Candida albicans, which is a common part of most people's microflora, but can cause it's an opportunistic infector or opportunistic pathogen. So it can invade parts of the body or be kind of recurrent or tend to overgrow. So some of you that are probably real tuned into microbiome and probiotics probably have thought about like yeast overgrowth and or fungal overgrowth. So that's one that can overgrow, but is part of most people's normal microbiome. And so we just have you kind of, list different characteristics that are here. Again, we're kind of just really leading up to the dichotomous key that's there. And make sure you use the pictures. Another note on the study guides, I'm seeing students insert their own pictures. Of course, I would love to see more hand-drawn pictures from everyone, and so we do ask you to draw, and most of you are doing that. If you have to pull something in from online, because maybe you just can't get it to draw up nicely on the computer, make sure you cite that and don't just stick a picture in there. So you'll notice the ones that we use, in the content file and things like that, you're going to see that, we mentioned where that came from in there. All right. So, just looking for some key features on uh, different microbes. This just has a little sorting and things like that. And this ties into this little table here, like looking at, they're having no nucleus, not eukaryotic, putting those all together there. So that ties into that first table. Um, I did have a question about on some of these where you have to fill in the blanks. Sometimes you just can't get it exactly the way it has it. This is not graded, so this is just for practice. So don't worry if you get something wrong or you're not able to, like, I think one time it needed a U for a micrometer, and it wouldn't take it. So, it's not for a grade. So it's just so you can practice those answers that are there. Okay. So you kind of go through the different features that are included in this lab, and this isn't really any particular group, it's just different groups of microbial features. Inclusion bodies, this is probably worth making a note of because you're going to see these two microbes in the final project. So this is Bacillus subtilis and Bacillus megaterium. Bacillus megaterium is typically called that because it has this pretty long, large appearance that's here. But they both contain inclusion bodies, so it'll look like almost like endospores, but not quite as swollen, but these kind of hollow or open areas inside of the cell. So that's pretty unique feature to both of those. It's a little more on inclusion bodies. So again, it's just kind of looking at different features. The flagella, we've worked on that a little bit already in one of the earlier units. Finding this fuzzy appearance in the periplasmic flagella versus one flagellum at either end when you have the amphitrichous arrangement. And then it's really important to start thinking about things like pathogenicity and virulence factors. This is something that comes up on the midterm and the final. So attributes of the microbe that enable it to cause worse disease or persist in the host to cause disease. So something that makes it either more difficult to fight off or able to invade host tissues. It gives it an advantage. So it's things like maybe having a capsule, having an endospore. Flagella sometimes is considered at least a pathogenicity factor because it enables bacteria to move through the host. So kind of go through those and practice those. And then we'll get on to the key. So I like that we have a video link here to a dichotomous key that maybe doesn't really have anything to do with our group. If you'll remember earlier in the term, you did a dichotomous key with, just things maybe you have at hand or on your desk. It's just important to use everyday familiar items at first to get familiar with what a key does. So here's a list of 7 organisms. We'll give you some pictures there to line up with those. And then you're going to start asking questions about those. Now, here they're presented as, a very flow chart, kind of yes or no. And then you would go to a certain step. Now, you can write your key out this way in this kind of flow chart type view. Later on, you'll have the opportunity to do kind of more arrows and draw things in. So, but some students stick with this format, this kind of yes or no. So it's important when you make your own keys later on that you make it in a way that makes sense to you that you can follow. That's the most important rather than following, to me anyway, than following a particular format. It has to make sense to, of course, the reader, but the user of the key. So you kind of go through, make sure you do your drawings that are in here. And again, if you've got to cut and paste something because you're doing it online, you can do something pretty crude quickly here with the drawing tool. But if you have to paste something in there, make sure you cite where you got that from. To me, sometimes it's easier to draw it than to try to cite it. So I'd rather draw it myself. Go through that, and then um, as you do that, you should start to find that you narrow things down. So the key to keys is that when you say yes to something, like let's go back to the very first question. So it asks if it has flagella or not. So when you say no, these first 2 items are now in their own group. So you can't regroup them later on because the way this key is set up, you separated them into bacteria with flagella versus or microbes with flagella versus microbes that don't have them. So once they're separated, you never merge the groups back. They stay separated. And then you continue to further separate these groups with questions until each microbe is in its own box or its own group. And that's the same principle you'll apply towards the end of the term. So as you see, as you get to the end, here's a different way to present that same information. I'm going to move this content file over a little bit here, make this more a feature of the screen. So this is the same questions about the flagella. Are there flagella visible or present? Yes. Now, what I like about this branch view is because you can really see how these 2 microbes, the Spirillum volutans and Proteus vulgaris, are in their own group, and you never merge them back, right? They are in the flagellar group. So then you ask one more question to separate those apart. And then in the no group, the organisms that don't have it, you continue to ask questions until every single microbe is by themselves. And you're going to use this approach for your final project. So we're just trying to get you warmed up. That is the main goal of this lab, is to just work on using, creating, and identifying using that dichotomous key to get used to that. Okay, that's all I have for this week. I will check in next week. We got more dichotomous key fun on the way, and we're going to be moving into more and more inoculations and seeing how those work. Lots of good videos and visuals. All right, have a great week.